Hello dear students, in this lecture we will discuss about another experiment to prove DNA as genetic material which was carried out or performed by the three scientists Avery, McLeod and McCarthy. It is nothing but a next part of or next level of experiment given or performed by the Griffith. In Griffith experiment we have observed that Griffith concluded that something is transferring from S type of cell to the R type of cells. That's why R type of cells become virulent, and that substance which is transferring from S type of cells to R type cells, it is nothing but the he called that as a transforming principle. He is not sure about whether it is DNA, RNA or other part which is transferring from S type of cells to the R type of cells. To move forward, these scientists carried out another experiment to prove that either is DNA, RNA or a protein which is transferring from S type of cells to RNA and R type of cells. He carried out another set of experiment, and the scientists are Oswald, Avery, McLeod and McCarthy. He carried out his experiment in 1944, provided the first evidence that Griffith transforming principle is nothing but the DNA, or the Griffith called that substance which is transferring from the S type of cells to the R type of cell, and that transforming principle or that information is transferred from S type of cells to the R type of cell is nothing but the DNA. That's why R type of cells become virulent and death of that particular mice. So to prove whether it is DNA or RNA or protein or other sort of substance transferring, he carried out another experiment in two different steps, so that according to the Griffith, the acute virulent pneumonia pneumococci was responsible for the Griffith transformation. That was in his experiment he used S type of heat-killed cells and live R type of cells, and after the death of mice he found live S type of cells or virulent organism cells he has observed, and he called that as a transforming principle, and according to that heat-killed virulent pneumococci are responsible for the Griffith transformation. So moving forward, these investigators who Avery, McLeod and McCarthy selectively destroy constituent in purified extract of virulent pneumococci using enzymes that will destroy DNA, RNA or protein. In other words, we can say, as Griffith said that something is transferring from S type of cells to the R type of cells, or something is taken up by the R type of cells to the S type of from the R type of cells from the S type of cells, whether it is DNA, RNA, protein or polysaccharide or other part. So to discuss whether it is DNA, whether it is RNA or protein, his or these scientists carried out the experiment and added, or he set this experiment according to that in each separate substance he added different enzymes which hydrolyze DNA, or you can say with respect to DNA he used DNase enzyme, or in case of RNA he used RNase enzyme, or for this the substances or the enzymes which degrade different substances to confirm that transforming principle is either DNA or RNA. So he carried out this experiment. They then exposed non-virulent pneumococcal strains to treated extract. Non-virulent, that is nothing but the R type of strains, and they exposed that R type of cells to the treated extract of DNA, RNA and protein. Transformation of the non-virulent bacteria was blocked only if the DNA was destroyed, suggesting that DNA is molecule responsible for the transformation. So when they mix the treated extract with the R type of cells, and they found that only the blocking, or we can say conversion of R type of cells into virulent strain become or found blocked only if the in case of that extract which was treated by the DNase enzyme. That means this DNA in the extract was destroyed. That means information is not transferred from S type of cells to the R type of cells. That's why R type of cells remain as it is, that is non-virulent. In the next slide we see he used three different or different sets of experiment, and he used R type of cells plus purified S cell polysaccharide. So in that they found that R type of cells remain as it is. There is no transformation of R type of cells into S type of cells. Next set of experiment they have used R type of cells plus purified S type of cell protein. They didn't know whether it is polysaccharide or protein or DNA or RNA, so that's why they carried out different experiments in different sets. So yes, cell protein, and in another step they also observed they see R type of bacteria remain as it is. Then third stage is use R type of cells plus purified S cell RNA, and that extract is treated with the RNase enzyme. So again they found R type of cells are remain as it is. Okay. In fourth stage they use R type of cells of purified S type of cells DNA. So they found that R type of cells are transformed into S type of cell. That means they sure about that the information is transferring from S type of cells to the R type of cell is nothing but the by means of DNA. So in next level they carried out S cell extract plus protease to degrade or hydrolyze the protein. They treat that extract with the protease enzyme plus R type of cells. They found that S type of colony is observed. That means protein is not a substance which is transferring information from S type of cells to R type of cells. In next step they use S type of cell extract plus RNase enzyme plus R type of cells. They found S type of colony. As again RNA is not a substance which is transferring R type of cells into S type of cell. In next step when they use S type of cell extract plus DNase enzyme plus R type of cells, they found that R type of cells remain as it is. That means the transforming principle is nothing but the DNA. Due to that something is taken up by the R type of cell, that's why R type of cells become virulent, and the transforming principle is nothing but the DNA. Hence, genetic information is stored or carried from the DNA or stored on the DNA. On that, transformation of the non-virulent bacteria was blocked only if the DNA was destroyed, suggesting that DNA is the molecule responsible for the transformation, or the Griffith transforming principle is nothing but the DNA, and hence it is nothing but the evidence about DNA is the genetic material which is responsible, which carry or store the genetic information, and due to that R type of cells are transforming into S type of cells. It is all about the experiment of Avery McNamara and McCarthy. Next part we will be discussing.